Thanks, everybody.
One of the sponsors of this speaker series, to whom we are very grateful.
Yeah. Well, good afternoon, everybody, and thank you for coming. And I guess I should say bon appetit as well. So before I start, I wanted to acknowledge my collaborators, some of whom are here. So Jessica Lynch Alfaro, she's at the Institute for Society and Genetics. Michael Alfaro, he's here at UCLA at the Department of Ecology and Evolutionary Biology. Tom Dial, he's an undergrad at UCLA, and he collected a lot of the data that I'm going to show about bats today. And then my colleague, Tom Eiding, he's at UMass Amherst. I'd also like to start by thanking not only the Institute for Society and Genetics, but NSF for funding that made possible this work, as well as collaborators and lab members who also helped throughout the development of the work that I'm going to present. So could we dim the lights a little bit? Maybe somebody could get it there. That would be great. Perfect. Yeah, I think that's good. Can everybody see it better? OK, so one of the central themes in biology is to understand not only the patterns, but the causes that drive biodiversity. And when we look at the diversity of organisms across the tree of life, there's one major pattern that emerges, and that is that neither lineage nor morphological distribution are evenly distributed across this tree of life. So take mammals, for example. We have some groups, for example, rodents up here, and then bats, that contain thousands of species, and they're very morphologically diverse. Whereas we have, in the other extreme, some groups of organisms, for example, this group here in white that contains elephants, which only have a handful of species, and they very much look like one another. And then we have other groups scattered throughout the tree that have different levels of numbers of species and morphological diversity. So a central theme in biology is to understand what are the causes that are driving these bursts in diversification. And I specifically focused on how the evolution of novel behaviors or novel anatomical traits might drive these bursts of diversification. Now, in the past, I've done this looking at how morphological traits in the skulls of bats might have led to their radiation into different dietary habits, as well as speciation rates. But today, I'm going to talk about an aspect of morphology that it's very obvious to all of us, and that is diversity in color patterns. Here are just a few striking examples of very beautiful color patterns in some vertebrates and invertebrates, frogs and birds, fish, and also insects. And this diversity in color patterns has been extensively studied in some of these groups, especially in birds. But we have less information when it comes to the evolution of color patterns in mammals. If you've taken a mammalogy class, you'll probably have been told that mammals are usually dull in color with a few exceptions. But today, I hope I can show you that that is not the case. There's a lot of interesting color patterns in mammals when we really look into them. So today, I'm going to talk about two main aspects of my research on color evolution. First, I'll talk about the drivers of diversity in facial color patterns in neotropical primates. And then I'll talk about the drivers of color diversity in bats. Two very different groups here. So I'll start with this work in primates, and I'll talk a little bit about faces. So faces are very interesting and striking and complex morphological structures, and they have evolved to have very important functions in social interactions in very visually oriented organisms. And these range a very wide array of species, including paper wasps, many species of carnivores, many species of birds, and of course, primates. When we look across these groups, I think we can all agree that primates probably take the prize in their diversity of facial color patterns, as you can see from this really small sample of different species of primates. So as an evolutionary biologist, I look at this diversity, and one of the questions that pops into my head is, what are the factors that are underlying this diversity? But in order to really understand and to answer this question, we need to think about what function primate faces are accomplishing. And it has been for a really long time that primates use facial cues during social interactions. They rely very heavily in this type of information. So they use facial color patterns, facial expressions, as well as different shapes of the face to get different types of information about identity, about behavioral intent, even physiological condition. Now, out of all these functions, conveying an individual's identity is a very important one, because we can think that this is actually the gateway to other social interactions. So we would expect that there are strong selective pressures on the evolution of facial patterns in order to convey an individual's identity. And in that sense, facial color patterns are thought to function in species recognition through a process that's called class-level recognition. And in that process, we have a receiver. Let's take, for example, this emperor tamarin here, who is surrounded by other individuals. It could be of the same species, of other species. And the way that he is going to identify individuals of his own species is by comparing the color patterns of these individuals to its own internal template, the template of its own species. And then that allows him to identify that particular individual, and then you can further interact socially or mate. So this is a very important function that is related to fitness, because this is what allows him to identify potential mates. And we can further exemplify that by looking at these different faces. And this is the same process, right? We would match each one of these faces to the pattern that we know it's a pattern for humans. And we can tell, well, this is a Wookiee, this is a Klingon, these are not humans, this is a human. Now, within a species, the facial color patterns are pretty much the same across individuals, for the most part for most species. I'm just broadly generalizing here. So it's more subtle variation within these facial color patterns, such as those seen here, that are used to identify particular individuals. So differences in different tones, in different parts of the face, differences in how morphological structures such as the eyes and the nose are shaped or distant from each other, will allow individuals to identify particular individuals within their social group. And this is the same type of process that we use to identify the people that we know, and this is the same type of process that facial recognition software uses to identify faces. So because of this, then, we can safely assume that because of this really important function of faces during social interactions and conveying identity, then social drivers might be very important forces driving the evolution of diversity of facial features. But what particular components of the sociality of species might be driving this diversity might depend on what kind of variation we're talking about. So here's what I mean by that. It might be that specific characteristics of the social system, for example, the pattern of migration, the hierarchy, and sex ratios, all of which could alter the dynamics within the social group, could create pressures for variation within species, as this could be used to identify individuals within the groups. Whereas, on the other hand, we could have that other factors, for example, the size of the social group, which has to do with the presence of conspecifics, or the degree of sympatry with closely related species, that is the presence of heterospecifics, could create pressures for interspecific diversity, which could be used for species recognition. So in this section of my talk, I'm going to focus on this part of the relationship, that is the social drivers of interspecific diversity of facial color patterns in primates. We also have to remember that faces, just like the rest of the color of the body, also have to accomplish ecological functions. For example, they have to allow species to camouflage against predators, or when their predators hide from their prey, and also facilitate other physiological functions, for example, thermoregulation. So we also wanted to look into how these different functions might play in the evolution of facial color patterns. So the overarching question that we wanted to answer was specifically, what are the social and ecological factors maintaining the diversity in the color patterns of primate faces? So that is the interaction among all these factors. Once we account for the evolutionary relationships among species, how much can we explain by the sociality and the ecology of species? And in order to look at these interactions, we tested two main hypotheses. The first one has to do with species recognition, in which we stated that diversity in facial color patterns evolved in response to selective pressures for species recognition. And we tested two main variables within this hypothesis. First, the size of the social group, that is the presence of conspecifics. So when we look at how social group varies across species of primates, we can describe or predict two different trends depending on whether species recognition or individual recognitions are emphasized. So let's take species that live in small groups or solitary as species evolve to live in larger groups. One scenario could be that the faces have evolved to be more patterned, to be more complex in these larger groups. And these faces that are more complex would be advantageous in these large groups because they could have more parts that can vary independently from one another. And this would facilitate inter-individual variation that could be used for individual recognition. So this scenario would emphasize individual recognition, an individual recognition function for facial patterns. Now, the other scenario could also be possible. And in this case, it would be the opposite trend. So that more complex faces would evolve in the species that live in the small groups or are solitary. And these more patterned faces could be advantageous for these species because they usually live very scattered throughout their environment. So these more patterned faces could be a louder signal of identity that could allow them to identify conspecifics, for example, potential mates, or to guard off other individuals that may be invading their territory. So in this case, there's more emphasis on species recognition if this is the trend that we found. We also tested then for the presence of heterospecifics in this relationship with facial color patterns. That is the presence of sympatric species. And we predicted that as species go from low levels of sympatry to high levels of sympatry with closely related species, then the facial color patterns are going to evolve to be more complex. And again, these more complex faces would have the advantage in these high degrees of sympatry of having more parts that can evolve independently from one another, in this case, fostering interspecific diversity that could be used for species recognition. Then we also tested an ecological hypothesis that facial color patterns are the result of selective pressures on ecological functions. And we tested some specific ecogeographical rules or predictions from these rules. And these are rules that have been previously tested in other groups of mammals, for example, carnivores. So we tested Glogger's rule, and this predicts that as species move from living in dry environments and more sunny environments to living in places that are more humid and more forested, they would evolve darker colors. And this has to do with an increasing need for thermoregulation, and also would allow these species to better camouflage in this more forested environments. Also, we tested the hair rule, which is an ecogeographical rule that has been less looked at in the context of mammals just in general. And this predicts that the thickness and the length of the hair is going to increase as species live in colder climates because this would aid in thermoregulation. And finally, for the evolution of eye masks, which are predicted to have evolved in several groups of mammals as a mechanism to reduce solar glare in sunny environments. So think about football players and baseball players that put the black paint under their eyes to reduce solar glare. So several groups of mammals have also evolved this strategy just with having a dark coloration around the eyes. So we tested this hypothesis using neotropical primates as a starting point. And here's just a very small sample of what these animals look like. As you can see, they have a very nice diversity in terms of the facial color patterns. And also, for our studies, they were really appropriate because they are a single radiation, so they're phylogenetically contained. And also, they're geographically contained since they span from Central to South America. And within this range, they span a wide variety of habitats, and the group as a whole varies a lot in terms of the social system, from solitary to groups of hundreds. Also, they have a high degree of sympatry with congeners. As you can note here, they are throughout the Amazon where very high levels of biodiversity. And also they have, as I mentioned earlier, a great diversity of facial features. Now, this creates a great opportunity in order to test our hypothesis, but it also creates some challenges. And that is how to quantify these facial color patterns in a way that's comparable across species that are as different as these. And biologists have developed really good methods in order to measure color, not only in mammals, invertebrates, and other vertebrates. But so far, we don't have a good way to measure patterns as a whole. So we have to come up with something. And as a first approximation, what we did was to create a measure that we call facial color pattern complexity. And I'm going to walk you through really quickly how we did this. It's fairly simple. So we collected photographs of adult males for 129 species of primates. These are both pictures that were taken by my collaborators, as well as pictures taken in captivity and in the wild that were in public databases. We selected pictures that were under good light condition and preferentially where the individual was facing the camera, like this guy. Then we subdivided the face into 14 regions, which are outlined here. And we selected these regions based on the variation in color patterns that we saw across neotropical primates. And then for each one of these little patches, we scored the coloration of the skin and the hair. And then we summarized that information by counting the total number of colors that were present across all these patches. So how many colors the face has. So this allows us then to create a continuous metric that goes from low levels of complexity, that is a face that is basically just one color, to high levels of complexity, that is a face that has many different colors. And it's a continuous metric that then we can use in our comparative analysis across species that have even very distinct color patterns. Now, to test the species recognition hypothesis, we ran phylogenetic generalized least square regressions. So what these are regressions that take into account the evolutionary relationship among the species. And the reason why we have to do this is because when we're running this broad comparative analysis, there is some of the variation or some similarities across species that are just due to their common evolutionary history. So we need to take out that variation and then see how much of the rest of the variation in the morphological traits is explained by the actual variables that we're testing. How much is due to adaptive evolution? So we ran these models by using the facial color pattern complexity values as the response variable. And then group size data collected from the literature as one of the predictor variables. And then data on the sympatry at the genus level per each species. And we calculated this by using geographic information system files and software that allows us to count the number of overlapping distribution at the genus level for each one of the species. And then these analyses were run in the context of a recent phylogeny to account for those evolutionary relationships. Then, to test our ecological predictions, we use our values of facial color pattern complexity, but also because Gloger's rule and the hair rule and eye mask make predictions about the darkness of the different facial regions, we quantified the pigmentation and the hair length for larger regions of the face. So across neotropical primates, we looked at which one of these smaller regions covaried mostly across species, and we grouped them into larger regions. And the reason we did this was to reduce the number of tests we had to run and also to facilitate our interpretations. So for each of these larger regions, we then quantified the pigmentation of the hair and the skin using these continuous scales that we created. And these were based on the variation in colors that we saw across species. And hopefully you can see these star colors here too. So it goes from basically white hair all the way to black hair, and then similarly for the skin. And then we did the same for hair length, going all the way from basically bare skin all the way to hypertricky or really, really long hair like this mustaches and the emperor tamarins. And then we put these data again into PGLS regression models where the facial complexity values or the pigmentation in each facial region or the hair length at each region were the response variables. So these were all separate regression models. And then here the predictor variables were the latitude and longitude at the center of the species distribution. So this gives us an idea of the environmental condition that the species are experiencing just on average. And then this, of course, was in the context of the phylogeny. OK, so now I'm going to show you some results. So this first figure that I'm showing you is phylogeny of neotropical primates, the one we use for our analysis. And the colors of the branches are representing the levels of facial color pattern complexity in this species. So we have warmer colors indicating higher levels of complexity. A face that has more colors is more patterned. And then in blue and cooler colors are the faces that are more plain. So also I'm showing here a few illustrations of neotropical primates just to show you what these levels look like. So one pattern that emerges is that first, well, we see that high levels of complexity have evolved in several different lineages, including owl monkeys, we have spider monkeys, and some species of tamarins. And then we have, on the other extreme, low levels of complexity in howler monkeys, wakaris, and then other types of tamarins as well. And then intermediate levels are kind of all over the place. But what is most important is that these patterns that we see in facial color pattern complexity actually seem to be related to the sociality of the species. So we found that facial complexity is related to social group size. And this is a negative relationship. So that the species that live in the small groups or are solitary tend to have the more patterned faces, and the species that live in the larger groups tend to have these more plain faces, just unicolored faces. So what explains this result? So besides these faces that are more patterned potentially being a louder signal of identity in this solitary species or small group species, complementing this scenario, it could be also that these more plain faces have some advantage for the species that live in the large groups. And here's potentially why. So we know from a lot of previous research that primates heavily rely on facial expressions to communicate. And this is an especially important form of communication for the species that live at large groups and interact at close range distances. And there is some evidence around some preliminary analysis with a little bit of data that is out there on facial mobility on primates, quantitative data. There's some evidence that species that have lower levels of facial color pattern complexity, the more plain faces, could actually be better at conveying information through facial expressions. And in fact, we see that the species that have the more plain faces have higher mobility scores than the species that tend to have the more patterned faces, which have lower mobility scores. However, the data is really limited. Quantitative data on mobility across neotropical primates and in primates in general is limited. So we really need more data in order to further test this hypothesis and see if indeed the more plain faces could be better at communicating with facial expressions. Okay, moving on and further supporting this hypothesis that facial color patterns function in species recognition, we found that the species that live in higher levels of sympatry, that is with a higher number of species that are closely related, do tend to have the more complex faces. So this supports this idea that a face has more parts, in biology we call modules, that could evolve independently from one another. This could foster interspecific diversity that could be used for species recognition. Now, even though, as you can see, we found a very strong effect of social variables on the evolution of facial color patterns, we also found a strong effect of ecological factors. So here's the map of distribution of neotropical primates. Hopefully you can see the outline here in black in Central and South America. And here just outlined some of the major biomes in South America. I'm going to show you a few cartoons that are going to illustrate how different parts of the face are changing. And the arrows are going to indicate what parts of the face are becoming darker. So first, we found support for Glover's rule. Remember, that's the rule that predicts that coloration is going to become darker in more forested and human environments. And this rule was met by the area around the nose and the mouth and the crown of the face. So these are species that live more westward and also towards the equator, especially in these Amazonian habitats. So that supports this idea that these darker colorations could actually be functioning in thermoregulation and camouflage in these habitats. We also found support for the evolution of eye masks as being associated with more sunny environments. So these eye masks tend to evolve more towards the east and also closer to the equator, all of which are more sunny environments. So these areas around the eyes could indeed be functioning in reduction of solar glare. And finally, we also found support for the hair rule, but only for the region around the face, so that is the face margin. So the hair around this region tends to become longer as species live away from the equator, that is in colder climates, supporting this idea that this can function in thermoregulation. Now, one important pattern that is also interesting to look from this data is that not all regions of the face are changing with the geographic distribution of species. And in fact, facial color pattern complexity was not related to the distribution of the species, which is the opposite of what we would expect if the coloration of the face was mostly driven by ecology, we would expect that the whole face would become darker as species live, for example, in forested and humid regions. But that's not the case. So this raises the interesting possibility that it is the interaction of social variables and ecological variables, which sometimes could create conflicting pressures, that are shaping this diversity of neotropical primate faces. Another interesting result that we found is that primate faces could actually constitute, we call in biology, many-to-one mapping, an example of many-to-one mapping. And many-to-one mapping is a general principle in functional morphology that states that a similar morphology across species, just by utilizing different parts of that morphology in different ways, could achieve the same function. So if you take a look at these two callitric species, if you pay close attention, you will see that one is basically the reverse color from the other. You see this hair tufts here are white, here they're black, the region on the face here is dark, here is white. But these two species actually have the same value for facial color pattern complexity. So this shows us how the same level of color pattern complexity can be achieved through different combinations of colors. So what this tells us is that species will still achieve certain ecological functions, for example, while still meeting the pressures due to their social interactions, which is achieving certain levels of facial color pattern complexity. And this is a very interesting result that matches this general principle that we see in functional morphology. So just to summarize this part, we see that there are social and ecological factors that maintain the diversity of facial color patterns in primates. So we see that after accounting for evolutionary relationships, we see a strong effect of functions such as species recognition that are shaping the variation in facial color patterns. But also we see an effect of ecological and physiological functions such as thermoregulation and reduction of solar glare that in turn seem to be affecting the pigmentation of different regions of the face. And it's the interplay across all these factors that then seems to be shaping the diversity of primate faces. Now, what we're currently working on is extending this work not only to other primates, but to other groups of mammals. And these are all groups that have very interesting variation also in their facial color patterns and in the social system. So we're looking forward to see how these trends that I just described for neotropical primates are met by these other groups. And if you're interested in hearing some preliminary results, we can talk about it after the talk. Okay. So now, for the second part of my talk, I'm going to talk about one of my favorite groups of animals, bats. So it's a complete switch of gears by switching to this group of mammals. So probably when you think about bats, you don't think about animals that are particularly colorful or striking in terms of their anatomy. But in fact, we have a lot of interesting color patterns and variation across bats. We have species that have spots. We have species that have crazy hair tufts, species with stripes, and many species also that have interesting facial color patterns. So I wanted to investigate then what were the factors that maintain this diversity of color patterns in bats. Now, asking this question in bats is a very different question than when we ask it in primates or even many other groups of mammals. And that is because of the way that bats interact with each other and with the world. They gather information mostly that is mostly auditory or olfactory in order to communicate or just to perceive their world. So we need to understand then what are the factors that maintain this diversity in color patterns in bats, and the role, for example, of social behaviors might not be as important, but instead it is the more ecological functions that could be then creating a higher pressure for this color diversity. Now, what kind of functions could it be, these ecological functions? So bats, like many other mammals, have very strong pressures to be cryptic, to hide from potential predators. And in fact, this is the general idea of for the evolution of color patterns in bats. And part of this is because, as you all know, bats forage and are active during the night, and then they are resting or roosting during the day. And during this daylight period, they're very vulnerable to predation by visually oriented predators. But not all bats use the same roosts, and not all bats have the same roosting ecology. So we need to think about then in what specific circumstances or roosting conditions bats are more vulnerable to predation. So I'll just describe a few types of roosts where bats would be more vulnerable to predation by visually oriented predators. So for example, we have many tropical species that make tents in the vegetation. So here's an example of a tent, and then if you look underneath, you will see bats like these or like these. So in these species, the male will chew on the leaves of this kind of palm-type tree, or also in the veins of these leaves, and then the leaf folds, and then the bats live underneath. And they're still visible for a visually oriented predator that's going underneath. There are other species that also roost in the vegetation, but in a different way. There are species that excavate roosts inside active termite nests that are arboreal, like this white-throated round-eared bat. So this is a colony of the bats when you look from underneath in the hole where the bats come into their roost. And then there are species that are even in more exposed situations. This is a picture I took in Costa Rica of a group of slack-winged bats. And I'll give you just a few seconds to see if you can find the bats. And if you can't, here they are. Here's the group of 5 bats here. And this is their natural roosting behavior. They're just basically on the bark of the tree. But as you can see, they have color patterns and then markings on their backs that allow them to be more cryptic even in these very exposed conditions. So this led us to hypothesizing that the evolution of pelage marking is associated with roosting ecologies that benefit from crypsis, particularly with these roosting ecologies in the vegetation. So we would predict that pelage markings, for example, counter shading, stripes, spots, and neck bands, would evolve in species that are particularly roosting in the vegetation. Now, where a bat roosts is important, but it's also important who is roosting with. So bats vary a lot in their roosting ecology in terms of the colony size. We have species that are solitary. We have only one individual roosting by itself. And then we have species that live in colonies of thousands or even millions. This is a colony of fruit bats in Australia. So if you think about it, the predation risk for an individual that is roosting by itself compared to an individual that is roosting in such large colony is not the same. The probability of being predated for each one of these individuals is lower than it would be by an individual that is roosting by itself. So we also wanted to test how the size of the colonies could have an influence in the evolution of body markings. So we predicted that species that live in the smaller colonies would have higher selective pressures for markings because they're at a higher risk of predation. Whereas the species that live in larger colonies would have a lower selective pressure for markings because their individual probability of being predated is lower. Now, another factor to take into account for the roosting ecology of the species is the actual size of the bats. So it's not the same to be a small bat than to be a large bat. Because if you're a small bat, there are more predators that can eat you. So smaller bats should have also higher selective pressures for these markings to be more cryptic. Whereas a larger bat has a smaller range of predators that can eat it. So they should have lower selective pressures for markings. So we would expect also that the presence of markings should decrease as body size increases. Now, to test this hypothesis, we collected data on the presence of pelage markings in bats. We did this for 914 species and collected the data from species descriptions and photographs. And we were able to classify their markings into 4 main types. So we have species that are uniform, and this is the majority of bats. And then we have species that have counter shading, so a darker dorsum and lighter ventral surface, species with neck bands, then species with spots, species with stripes, which could be located in the body, in the face, or both. And then we collected data for the roosting ecology, also from the literature and species descriptions, and we classified them into the 4 main types that we were able to find. So we have species that live in the exposed vegetation. So these are species that are roosting in the surface of trees. We have species that are hanging from tree branches with little or no coverage from leaves. Then we have other species that are living in the vegetation, but they're more concealed. So the species that live in the tents that I described earlier, species that live in the leaf litter, and if you can find a bat, it's here. So it's very well concealed, and it's matching its background very well. And also species that live in hollow trees. And then the other types of roots included rock crevices and caves. And along with characterizing the species in terms of what types of roots they were using, we also collected data on the size of their colony and on their body mass. And we put this data again in these PGLS regressions that accounted for the evolutionary relationship across species. And here, the response variable was the presence or absence of the different types of markings. And the predictor variables were the roost type, which is a categorical variable, and then the size of the colony, which is a continuous variable. Also the body mass. And I should say here that these two variables are actually positively related. So the species that live in larger colonies also tend to be larger in size. So there's a parallel here in terms of reduction of risk of predation. So we didn't include these two variables in the same model because it would break the rules to conduct our regression. But when we test separate models for these, we obtain the same results. So here's the phylogeny of the different families of bats that we study. So in this case, I collected the branches in this phylogeny. So I'm showing you families of bats, not species of bats. And highlighted are the ones where we find marking, for example, countershading, net bands, spots, and stripes. And then I also wanted to show you the kind of taxonomic coverage that we were able to get during this study. Here is the sample sizes that we examined in terms of numbers of species, and in parentheses are the total number of species within those families. We had a really good taxonomic coverage that allows us to really investigate the evolution of these patterns. Okay, so moving on to this first hypothesis, how do markings evolve in terms of the occupation of vegetation roosts? So this table that I'll show you has, there are the different marking types, and then all of them combined. And then we're going to see the slopes here with the occupation in different types of roosts. So these are all vegetation roosts all combined, the exposed vegetation and the concealed vegetation. And you're going to see there are some numbers that are bold in red, and these are the ones, the slopes that are significant. So we see that across all vegetation roosts, we find that all markings in general tend to have evolved under these conditions. So bats that live in the vegetation tend to have markings. But this relationship is mostly driven by the presence of stripes and neck bands. Now, if we look at the exposed vegetation, is a very similar pattern. And then in the concealed vegetation, it's also very similar. We lose some significance here for the stripes, but this is mostly because the sample sizes there get really, really small. So we have then a lot of evidence then that it's mostly stripes and neckbands that are evolving in the vegetation. So why are these two types of markings advantageous for these bats? So stripes are a really classic example of disrupted coloration in mammals. This is the typical explanation of why zebras have stripes. They break down the body of an animal, so it's harder to pick up individual animals, especially when they're all together. So it's harder for a predator to target a particular individual. And this has also been described in insects and other animals that also have this kind of disruptive coloration. So the same could be said for bats. Remember I was showing you the tent-making bats, and when we look them from below, they would look like this. So you see this very striking facial marks here. Also, you can see the ones from their back. So this would create also disruptive coloration that would make it more difficult for a visually oriented predator to pick up particular individuals who actually recognizes as a bat. Also, some of these markings could also mimic the patterns of lights and shadows that sometimes are coming from the little holes in the leaves that they're living. So it's not only disruptive coloration, but it could also be a little bit of background matching too. How about neckbands? So neckbands are also a very interesting type of markings that bats have, and we think that this could be a particular type of countershading that is unique to bats. Now, what is countershading? So countershading is a pattern that it's been observed in many groups of vertebrates and even invertebrates, and it's characterized by having a dark dorsum and then a light ventral surface. And this helps organisms to be more cryptic because when they are seen from below, they're seen against a light or a bright background, so they're more cryptic. And when you see them from above, then they're against a dark background, so again, they're blend more with their environment. And even when you see them from the side, if the animal is standing like this, it might create a shadow on the ventral part of the body, which could also help it be a little bit more cryptic. Now, if you look at all these species that I'm showing here, and for most species that have counter shading, their locomotion and the way they sit and stay is horizontal, or I should say parallel to the substrate where they locomote. But that is not the case for bats. Bats, when they're roosting, they're hanging upside down. So this type of counter shading wouldn't really help in this situation. What could help would be a type of coloration where, again, the bottom is still light, and then the top is still dark. But in this case, because of their roosting behavior, this turns out to be the anterior part being light and then the posterior part being dark. So it's a special type of counter shading that is unique to bats. Now, moving on, now I'm going to show you some results about how the evolution of markings might be related to other aspects of the roosting ecology, that is, their colony size. So in general, we found that as colony sizes become larger, we see that there is a drop in the presence of markings in species. So species that live in larger colonies seem to have lower pressures for the evolution of markings, just in general. So this matches very well our prediction. But let's see what happens when we break down this into the different types of markings. So when we look at spots and stripes, this still follows a trend. When we look at neckbands, then we get into trouble because the slope is actually positive. So what's going on here? So I'll start with the easy ones, the spots and the stripes. We see that, again, less color markings in species that live in these larger colonies. And this could be in part because these species are the ones that tend to live in these tents in the vegetation. And these type of roosts would create constraints, actually, in the size of the colony. There are only so many bats that can fit under a little leaf. So there could be parallels between the size of the colony size, which is constrained by space, and then pressures for evolving these types of markings to be more cryptic. Now, this could also explain why neckbands would have a positive relationship with colony size. And this is because most of the species that have the neckbands tend to live, so there are high pressures for spots and stripes, and colony size is limited in these species. So in neckbands, most of the species that have these neckbands tend to live in this type of roost, very open environments. There is little or no limit to how many bats can roost together. So we can see then that will be a tendency towards having larger colonies, but still there will be pressures for evolving these type of neckbands because of pressures of being more cryptic towards predators. Now, one aspect that I haven't talked about here with bats, and that is particular for these type of bats, and I should mention these are fruit bats, megabats, is that they could also be using this marking for some sort of social communication. And for most species of bats that are not visually oriented, this might not be possible. For these guys, they have really large eyes. So they could actually be using some sort of color signals also to recognize individuals or to interact socially. This is an epaulet bat from Epomophorus. And they have this particular displays where they have these tufts of hairs that rest into pockets, and they can avert them and do this displays to females. So there's still room there for some social pressures also driving some of the evolution of these color patterns. But still, we need to document more of these types of behaviors. So just to summarize and to really drive this point home, we see that then the diversity of color pattern in bats is not only then explained by the evolutionary relationships across species, but we see that there are important ecological pressures for the roost type or the body size that the species are utilizing, as well as the size of the colony, all of which then seem to be shaping this diversity in the color patterns of bats. And now, just to wrap up, I hope that with these studies in the facial color patterns of primates and the the body colors in bats, I have been able to illustrate this type of interactions among ecology and behavior, and how actually behaviors can be a really strong force in driving the evolution of morphological diversity. And with that, I think I'll stop there and I'll take any questions.